Um, Thank you all for coming. It's my great pleasure to introduce Dr. Nan Yan from Immunology. Nan is now the vice chair of Immunology, and from his CV, we can tell that he will be promoted to a full professor in the fall. Congratulations, Nan. Nan is a well-recognized immunologist, although he just joined UT Southwestern about 10 years ago. So he was the recipient of the prestigious Boros Wellcome Award in Pathogenesis. And now he has 4 NIH grants, including 3 R1s. So therefore, he's one of the researchers on campus who received more number of NIH funding. Nan obtained his bachelor from Fudan University in Shanghai in '99. Then he came to the States to pursue his Ph.D. in molecular biology with Paul McDonald at the UT Austin. After getting his PhD in 2006, he moved to Boston to receive a postdoc training with Judy Lieberman in cytotoxic T cells and antiviral immunity at Harvard Medical School. Then in 2011, Nan became assistant professor and endowed scholar at the UT Southwestern. Nan is working on innate immunity, and he identified novel immune mechanisms in various types of disease, including infectious disease, autoimmune disease, neurodegenerative disease, and cancer. In particular, mechanistically, Nan is very interested in the STIM pathway and developed new tools and identified novel mechanisms for STIM trafficking and signaling. So when people talk about STIM functions, most are about the induction of type 1 interference. Nonetheless, Nan discovered certain STIM activities that are independent of interference. So how did he find this and how does this work? Nan will talk about in today's seminar. So let's welcome Nan for his talk, STIM trafficking and signaling in health and disease. Nan, it's yours.
Yeah, thanks, Alec. A very nice introduction. So it's my great pleasure to give my seminar today. So I'm going to start with the overview of the different things we do in my lab. So my lab is mostly interested, like Alec says, most interested in innate immune signaling. So I have a big cell in the middle of your screen. So the majority of the innate immune cells have this built-in sensing networks for detecting, mostly evolutionarily for detecting microbial pathogens. So we have the traditional toll-like receptor TLRs either on the cell surface or in the endosome, detecting viral, bacterial proteins, nucleic acids. We also have cytosolic DNA sensors and RNA sensors. And when these innate immune sensors all get engaged, most for microbial pathogens, they will trigger a very strong immune response. We're interested in one type of immune response, interferon response. And some of the early work in my lab has to do with how viruses or bacteria are interfacing with the innate sensing pathways, how they evading or antagonizing these sensing pathways. My lab is also interested in autoimmune disease related to these pathways. Apparently, a number of the key regulators of these pathways, when you have genetic mutation in those regulators, then you end up with chronic activation of these signaling that will lead to a variety of different autoimmune diseases. One particular group of autoimmune disease called interferonopathy has to do with the chronic interferon activation. In more recent years, we also become into the cancer immunology and the neuroinflammation that are also related to some of these innate signaling pathways. So one of the particular molecule we spend a lot of time looking at is the STIM pathway. So I'm gonna touch on essentially all 4 corners of these disease relevance of STIM pathway. So to begin, so STIM is an interesting molecule. It's a ER anchored transmembrane protein. It sits on the ER as a dimer, and traditionally when STIM gets activated from receiving a cyclic dinucleotide, So the secondary nucleotide coming from microbial infection. Either it's a retrovirus like HIV that will introduce RNA, reverse transcripting DNA, or it's a DNA virus or bacteria introduce the DNA. Any of the DNA component will be sensed by C-gas, which will subsequently produce CGM. That's a ligand that will activate STING. Some of the bacteria will also produce secondary nucleotide on their own, like secondary AMP or GMP. That can also directly activate the STING. So in autoimmune diseases, if you have a genetic mutation in the nucleuses, especially DNAs, like TREX1, DNase2, that will cause accumulation of self-DNA that will trigger C-gas and then STING. Mitochondrial damage will produce DNA that will trigger the same pathway. In more recent years, there's also gain-of-function mutations being identified in STING protein that will chronically activate the protein that leads to autoimmune disease. So one of the things that sets STING pathway apart from many other innate signaling pathway is there's an interesting component of trafficking of STING molecule. A lot of innate immune signaling pathway, you are essentially looking at a cascade of molecules and that are essentially relaying the signaling from one molecule to the next. But STING pathway is different in the sense that immediate after activation of STING will go through a fairly interesting trafficking process. I'll go into details later. And that's important for both turning on and then turn off the signal and eventually rendering the signal. So in terms of therapeutic translation, one of the most exciting angles of therapeutic translation is cancer immunotherapy. Now, almost every pharmaceutical company now has a STING on their portfolio, mostly producing STING agonists to trigger potent interferon signaling. So once you activate STING, so the hope is you activate the antigen presenting cells like dendritic cells that will prime T cells to get better anti-tumor response. So this is some more details about STING trafficking and signaling, about both the mechanics of it and the spatial control of this process. Excuse me. So like I said, STINGs start on the ER, and the early structure studies suggest that STING sits at the dimer is the butterfly-shaped dimer. As soon as it binds a ligand, and the structure will changes a bit and stands up. And then the Crow-EM structure from James Chen's lab shown that it's also go through a 100-degree rotation that will likely trigger the STING to exit from the ER. That STING will traffic to this ER Golgi intermediate compartment in the Golgi, where it recruits the TBK1 kinase and then IR3 transcription factors. So there are also Crow-EM structures supporting that. So the TBK1 recruits the STING oligomers, and that will cause the phosphorylation of STING itself, phosphorylates TBK1, and then recruits IRF. This is a transcription factor, eventually goes to nucleus and then turn on the interferon gene programs. So then the trafficking doesn't stop here. And the STING will keep moving on to what's known as post-Golgi vesicles. The identity of these vesicles are much less understood, and it eventually gets to lysosome, the STING protein gets degraded. So during this process, the signal turns on once it gets to Golgi, and then turns off once it gets to the lysosome. So it's a fairly dynamic process, and it's also very well regulated. And the process after Golgi, this post-Golgi phase of these processes are not very well understood. So this is a quick movie about STING, just to give you a sense of what does it look like. So you're looking at STING GFP that at steady state, it looks like ER. So it has this paranuclear ring and then a vascular look inside of the cell. Once you trigger STING trafficking with the ligand, you can see this thing quickly moves to this Golgi, condenses on the Golgi, and then goes past Golgi into these vesicles. And if you follow this movie long enough, this is all within hours. If you follow this movie for a few more hours, then all these GFP signal will be disappeared. So the protein get very quickly degraded. So our first entry into the STING biology is from studying a gene called TREX1. So TREX1 is also known as DNase3. So it's a celloplasmic DNase. And it was initially found to be associated with a rare autoinflammatory disease called Aicardi-Goutieres syndrome. So this particular disease mostly affects newborn infants and young children. So the children have this brain calcification that's mimicking congenital infection, but eventually they find this is the genetic disorder with chronic activation of the type of interferon. So these patients have classical the interferon signatures, have skin lesions, resemble some of the tuberculous lupus. They have elevated autoantibodies. And one of the first genes that's been associated with disease is TREX1, also known as DNA3. So over the years, there's a number of other genes that have also been associated with the loss of function of RNase H2, loss of function of our RNA editing enzyme ADAR1, and the gain of function of MAVs. So if you look at this picture as a whole, that really what's causing this Aicardi-Goutieres syndrome is a dysregulation of nucleic acid sensing. So this is a simplified diagram of nucleic acid sensing in the cells. I have DNA sensing on the left-hand side that goes through cGAS-STING pathway, and RNA sensing on the right-hand side that go through the RIGA-MAVs, and RIGA-MD5 as an RNA sensor, and MAV as an adapter. Eventually, both pathways feeds into activating TBK, IRF, and the interferon signaling. So, so TREX1 has been one of the prime example of this particular disease. And over the past few years, that the genetic understanding of the TREX1 disease has been very well worked out. So now we know that one of the reasons the TREX1 knockout mouse recapitulated the human disease very well. So they also get systemic autoimmunity and then early mortality. So if you delete cGAS, all the TREX1, take a TREX1 knockout mouse. So the black line is the survival of TREX1 knockout mouse. You can either delete 1 copy or 2 copies of cGAS, you completely rescue this mouse. The same thing's the STING, the black line TREX knockout, you delete either 1 or 2 copies of STING, you completely rescue the disease. Interferon receptor, 2 copies of interferon receptor, you rescue TREX1 disease. IRF3, if you delete either 1 or 2 copies, you rescue the TREX1 disease. So that tells us that this is essentially maps out this cGAS-STING interferon pathway as being almost the singular pathway that drives TREX1 disease. So this gives us, important evidence for therapeutic targeting. Now that there are different groups that are developing either cGAS inhibitors, STING inhibitors, TBK1 inhibitors. We already have JAK1, these Zhang kinase inhibitors for the interferon JAK-STAT pathway that can treat this particular disease. So we've shown a few years ago using one of the TBK inhibitors that are initially developed by Mike Y here for cancer that show that if you treat a TREX1 mouse with TBK inhibitor, you can actually rescue the disease quite a bit. More recently, we're also essentially using TREX1 knockout mouse as a model to test the pathway inhibitors. One of the STING inhibitors we recently tested also is remarkably effective. You took one of them, so here's the mouse that is a mock-treated TREX1 knockout mouse. And then these are the 2 STING inhibitor-treated mouse. Just after a few weeks, the mouse looks dramatically improved after you shut down this pathway. So this has also translated back now to the patient. So the Zhang kinase inhibitors has been used in treating some of our Aicardi-Goutieres syndrome patients, has been fairly successful. This is one study in New England Journal last year where they treat several patients with Zhang kinase inhibitors, and then the exterior pathology has been drastically improved. The developmental skill is indicating of the neural pathology associated with disease are also fairly well improved when you target this pathway. This is another example of a tuberculous lupus associated with TREX1 mutation for targeting the interferon pathway. They also drastically improve the tuberculous lupus. So this is, this is in terms of cGAS-STING pathway driving autoinflammatory disease. This is one of the success examples about as long as we understand the cascade or the genetics of the pathway using mouse models, we can fairly successful treating this pathway. So then in 2014, there's a number of mutations that have been found associated with the STING gene. So when these mutations were initially found, we thought that we know these pathways so well from studies from TREX1 and other cardiac tear syndrome, and we should immediately know how to treat this. But one of the interesting findings one of the interesting clinical findings from these patients with STING gain of function mutation actually develop a very different clinical profile compared to the TRAX1 patient. And the most striking profile, these patients are suffering mostly from pulmonary disease. So these patients, they have some systemic inflammation in terms of fever, but the majority of the patients will develop pulmonary manifestation, either interstitial lung disease, lung fibrosis. They also have T-cell cytopenia. So these are the clinical features that we're now seeing with the classical type 1 interferon or cardiac tear syndrome. So that puzzles us, and then people in the field to understand why do a STING gain of function mutation are different from a chronic activation of the STING pathway. So it tells us maybe there may be other activities of the STING are actually mediating some of this. So just to show you the mutations. So STING, this is it's a crystal structure of the STING dimer. The pocket is where the ligand binds to. These gain of function mutation is actually at the base of the structure that are not affecting the ligand binding. So we in collaboration with Jonathan Miner and the WashU, so we made a STING gain of function mouse. We introduced the same mutation into the mouse, and then remarkably, mouse also developed lung disease. So develops apparent vascular infiltration in the lung, and then most of these mouse also have early mortalities. We also did immune cell profiling of these mouse, and then we also find T-cell deficiency or T-cell cytopenia in these mouse. So this is a site of the spleen where you see CD4, CD8 T cells, and in the wild-type mouse, but in the STING mutant mouse, we're losing most of these T cells, but we're gaining the myeloid cell population. So we're seeing a similar T-cell cytopenia in the mouse. And what's even more surprising on this is this whole clinical disease is independent of cGAS or interferon activity. So once we have the mouse, you can do genetics. So you have the gain of function mouse, and you can cross this mouse to either cGAS knockout background, IRF knockout, or interferon receptor knockout. And we see the same lung disease, the same T-cell cytopenia. So this, again, reinforces our thinking that the STING is likely driving other activities. So this is actually will be the first set of physiological evidence that the mammalian STING can drive a cGAS interferon independent activity that leads to clinical disease. And this actually opens up a whole new can of worms that we're seeing more and more evidence of this later on. So in terms of molecularly, how does this work, right? So we first looked at where do this mutant localize to. So here's looking at wild-type STING, it localized to the ER. And when we look at this point mutation, gain of function mutation of STING, they actually localize to this ER Golgi intermediate compartment called ERGIC. So they constitutively localize to the ERGIC. We think this may be one of the reasons why these mutants are constitutively activated, because we know that when STING's on the ER, the signaling is off, and when it goes to the ERGIC, the signaling is on. And regardless if it goes to ERGIC because of ligand binding or because of other means. So another thing we looked at, this is actually working with collaboration with Jen Liu in your department that help us with characterizing some of these chasm effect. So we thought we should look at T cells because the T cells are really diminished in these mutant mouse. So, but there's still some. So we took out these wild type and the STING gain of function mutant T cells, and then just take a look at them. And apparently these gain of function T cells are actively undergoing apoptosis or cell death when you pull them out of the mouse. And these also T cells are actually also chronically activated. And by looking at a variety of different cellular pathways in these T cells, what we eventually find is these T cells are having very robust ER stress and UPR being turned on in these T cells. And they have severe chasm problem as well. And it triggers a cell death in these T cells. And if you isolate a macrophage from the same mouse, the macrophage is perfectly fine. Somehow these gain of function activation of STING only causes the problem in the T cells. So I think, there's still more we need to do in terms of fully understand what causes this STING gain-of-function disease and why that's different from the canonical STING interferon-driven disease. But I think we have enough understanding that that tells us something's very different. I think at the very basic, we think the activation of the STING pathway in the macrophages predominantly gives you cause interferon response leading to inflammation. And these type of disease can be successfully treated with JAK1 inhibitors. But these gain-of-function mutation somehow actually activates STING in the T cells. And then when you activate STING in the T cells, you rapidly causes cell death. And that somehow leads to T cell cytopenia and lung disease. How that causes the lung disease, we still don't quite understand. And a lot of the lung pathology actually looks quite similar to these SARS-CoV-2 infection, and that's another interesting feature about what is STING doing in the lung and then what are those functions are. In terms of therapeutics for the SABI patient, the JAK1 inhibitor has been tried on SABI patients. It's been not very successful, and we think that maybe we should target something entirely different. And we show in the mouse at least targeting ER stress and UPRs are effective at treating these SABI patients. So, from that point on, we become even more interested in what are the other activities that STING has, and what are these interferon-independent activities, and what are, especially their physiological functions. So, if you look at the evolutionary history of STING gene, they actually tells you that the interferon activity of STING is actually a fairly recent event. So this is alignment of STING gene across evolution. The interferon domains only in these green bits of C-terminal tail, and that's only present in mammalian STING and some of the vertebrate STINGs. During early evolution, the STING proteins largely conserved, but they don't have these interferon domain. And these species don't have the interferon genes. So this tells us that perhaps there are other functions of STING that's actually fairly evolutionary conserved throughout, and then just the mammals that will start seeing the interferon activities. So in order to know what are all these interferon independent activities, so we decided to make a new mouse. So we made a point mutation, 365A. So this is a serum residue that's critically required for STING to activate interferon signaling. And if you make this point mutation, potentially you will be able to preserve everything else that STING able to do, and this molecule will just not able to do interferon. So now we have 3 mouse. We have a fully capable STING molecule. We have STING deficiency that lose all the activities. Then we have a molecule that will trigger all the interferon independent activities and lose just interferon. So this is the validation of the mouse. Show the mouse really, that mutation precisely deletes just the interferon activity. If you look at this Western blot, the different STING signaling, downstream signaling activities, in the wild type, we're turning on interferon signaling and NF-kappa Bs. Autophagy is discovered by James Chen's group. And the STING deficient mouse loses them all. And the 365 mouse only loses the phospho-IRF3. And it still have NF-kappa B, autophagy, and many other activities. And if you look at interferon cytokines secreted in this mouse, the blue is the interferon dead 365 mouse, and we're not detecting any interferon that produced in this mouse, even when you challenge with DNA or CGAM. But other cytokines, IL-13, the GM-CSF, secrete just fine. So we think these molecules, 365, are perfectly functional with just not interferon signaling. So the first thing we did is to challenge these mouse with the herpes simplex virus. So this is the DNA virus, has been one of the go-to virus to study the STING pathway. It's been previously shown that the STING deficient mouse are highly susceptible to these infections because it's unable to recognize the DNA and then produce antiviral response. So we're curious whether this interferon dead version of STING, how much antiviral response we're getting out of this. So surprisingly, the wild type mouse controls the herpes virus fairly well, and the STING knockout mouse succumb to infection very quickly. So you don't have fair defense. The 365 mouse actually control the virus also fairly well, fairly close to wild-type mouse. So I think this tells us that whatever the antiviral response mediated by stain is actually independent of interferon. And the same observation has been made by both Russell Vance's group and the James Chen's group. And we don't know what these interferon activities, independent activities are, but we think that's something quite interesting. Another thing we found from looking at these mouse is that the macrophage and STING drives a very different signaling program in macrophages versus T cells. So if you just look at, so we have 3 mouse, we can do pairwise comparison to determine either interferon-dependent genes or independent genes. In a macrophage, where STINGs really only regulate the predominant interferon-dependent genes. And another interesting thing about macrophages is not just increasing or the turn on genes, there's a vast amount of program that actually turn down expression of these genes, and we don't really understand what those genes are. But in the T cells, and STINGs predominantly regulate the interferon-independent genes. And if you do the pathway analysis, many of these are these classical T cell signaling pathways, like cell death, TCR signaling, and NF-kappa B signaling, and etc. So one of the particular signaling pathway interferon independent activity is cell death. So this is a simple experiment that demonstrates that. So if you take the macrophage and then the 3 different genotypes of macrophage, the wild type STING knockout or the interferon dead version of STING, and trigger is the cell permeable STING agonist. With the high dose curve, we're not seeing any cell death in the macrophages. But if you do the same thing with T cells, and then both the wild type and the interferon dead version of the T cells rapidly commit cell death, and with a fairly low dose of STING agonist within 24 hours. And it's only the STING knockout T cells are protected from the cell death. So we start thinking, what is the physiological function of the STING-mediated cell death, besides what we mentioned earlier, causing the SAVI disease? We thought this may be something could be used for in a cancer setting. So we designed a set of cancer experiments to test this. One of the thinking about cancer, especially solid tumor, is that the solid tumor, because they have so much replication stress, the solid tumors could be secreting either tumor DNAs or CGAM to the tumor microenvironment. And these DNAs or CGAM can then activate other immune cells in the microenvironment. So the experiment we set up is we will put in a B16 melanoma tumor in the T cell deficient mouse, in the rag mouse, and then we'll adoptively transfer into these either wild type or different genotype of T cells and ask the question, how do each one of these T cells control the tumor, right? So what we find is if you don't have any T cells, the tumor grows fairly rapidly in these mouse. If you put in a wild type T cell, these T cells can control tumor to some extent. But if you put in the STING knockout T cells, these T cells control tumor much better than the wild type T cells. So we think the reasons being that these STING knockout T cells are protected from this cytotoxic environment in the tumor. So one of the supporting evidence of that is if you actually isolate this tail from these tumors and then quantify them. If in a wild type scenario, you always see a large percentage of tails are undergoing cell death. But in a STING knockout T cell isolating from the tumor, there's a much less percentage of them are undergoing cell death. So we think the difference is here in what we call the CGAM susceptible tails. And here's either looking at percentage cell death or the tail numbers isolated from T cell. We think there's a substantial amount of tails that actually are undergoing CGAM or STING-mediated cell death in the tumor microenvironments. So that will be an interesting future direction to figure out what these tails are and what are the properties of these tails. So, I think in terms of the anti-tumor activity, we think the STING can actually mediate both anti-tumor and pro-tumor activities. And this is something important to keep in mind, especially when we're thinking about targeting the STING pathway for tumor therapies. We think when you activate STING in myeloid cells, antigen-presenting cells, and K cells, and you're getting inflammation, and you're definitely getting anti-tumor activities. But we think because STING activation T cells causes such a potent cell death response, and we should be really careful about thinking about administering STING agonists, for example, in the tumors. And then that has already been seen with some of the early clinical trials. Like the Adora's compound early on they had a study showing that if you do the high dose STING agonist, and they will cause massive T cell death in the tumor microenvironment, and then the loss of anti-tumor immunity. So we think, even the homeostasis for solid tumors, that when you're secreting either DNA or CGAM to the microenvironment, and these secretion could also cause us both the either anti-tumor response or pro-tumor, there's a tumor evasion, immune evasion type of response. So another thing like I mentioned before, we've been really obsessed with STING trafficking. We really want to know that what is happening when STING traverses through the secretory pathway, right? So it starts from ER, goes to Golgi, vesicles, and lysosome. And we think this is probably not a passive process. It's probably a highly coordinated process. One of the things we thought we should do is it would be really nice to actually take a snapshot of each location when STING gets there, and then see what's around it, right? And then what are the molecules that's hanging out with the STING in that particular organelle, and then what do they do in terms of regulating the STING function. So this is one of the recent unpublished studies. So what we did is do a proteomic STING cofactor screen. And so the flash, the stem shot, so we used a STING fusion protein with APEX2. Essentially, APEX is an enzyme that allows short range labeling of biotenylate, their near neighbor protein. And you can flash the cell with this hydrogen peroxide to transiently label the neighbor protein, essentially taking a snapshot of that particular time and location. And then you do this at different time during STING trafficking that we know, STING's either on the ER or the Golgi or vesicles or lysosomes. And we actually end up, pull down to do biotenylate pull down, do mass spec. We end up with a number of interesting cofactors in each one of these locations. So one of the cofactors I'm going to talk to you guys about is the NPC1. It's one of the protein we found on the lysosomes. That's turned out to be a really interesting protein. So some of you may be familiar with NPC. This is associated with Niemann-Pick disease type C. So the biochemistry of the NPC has been largely worked out by Brown Goldstein's group, and then also has to do with the cholesterol egress biology. So both NPC and NPC1 and NPC2 are required for cholesterol to egress from the lysosomes. And NPC1 is being a lysosome transmembrane protein, and NPC2 is a luminal protein. And when you have defect in either one of these two molecules, you ended up to accumulate accumulate cholesterol in your lysosome, and then you deplete cholesterol from the ER and other part of the cell. So that's the cellular defect. And then that cellular pathology will cause the neuronal damage. And that's leading to neuropathology mostly in the cerebellum. And there's very good mouse models that recapitulating this. And eventually, this NPC deficiency will cause a neurological symptom involving motor and coordination defect in the humans and also in the mouse, and leads to premature death of the humans. The immunological feature of this NPC disease is actually not quite well understood. Only a couple of years ago, that when people looked at the whole brain lysate from the NPC1 deficient brain compared to the wild type brain, they found that the NPC1 deficient brain had this type 1 interferon gene signature that they thought was interesting. But nothing else was really known to be involved in driving this particular disease. So we thought we should start with cells. So we start making a CRISPR knockout NPC1 in the mouse embryonic fibroblast. So we just look at these fibroblasts at a steady state, look at the baseline interferon gene expression. So what we call the interferon stimulated gene, ISGs. So we look at throughout these ISGs, and then using 2 independent clones on MPC, they all chronically elevated when you knock out the MPC gene. So this are consistent with the previous observation in the mouse, that when you delete the gene, you get a chronic activation of the type interferon signaling. And if you challenge these cells with a STING agonist, Dmax, Dmax AA, you also get a much more robust type interferon response compared to the wild type cells. So this signaling is STING dependent because when you have the MPC STING double knockout cells, then you lose all that ISG expression. You look at MPC CGAS double knockout cells, you're not losing these ISG expression. So that tells us that somehow in MPC deficient cells, STING is activated, chronically activated, independent of CGAS. We also went one step further to show this is even CGAM independent. So this is the heat map, essentially looking at a list of ISG, this is an interferon gene signature. In the MPC wild type cells, you see very low expression. MPC with knockout cells, you see much elevated expression of these ISGs. In MPC STING double knockout cell, you lose all the expression. Most of these are STING dependent. When you put back wild type STING, that drives the expression of these genes. When you put back a STING mutant that unable to bind CGAM, you still drive the expression of these genes. So we think really this interferon gene signature or inflammation are driven by STING, but somehow independent of CGAS and CGAM. So in terms of the mechanism, let me give you the model first, and then I can show you some supporting evidence. We think because we discovered this proteomic screen on the lysosome, we think really the MPC1 is the lysosome adapter for STING degradation. We think in the MPC1 deficient cells or deficient scenario, we're missing these lysosome adapters. So that causes a STING to accumulate and then continue to signal. But then this is actively independent of CGAS and CGAM. We really, we think the STING is actually activated by a trafficking mechanism that we call this tether trafficking by SRBP and SCAP complexes. And this is causing so trafficking causes the prime of the signaling, then loss of the lysosome degradation causes the boost. So we think this is the prime boost model that drives very severe neural inflammation. So let me show the SRBP priming. So in order to show that, so we have two different scenarios. We have a MPC knockout scenario where the SRBP is chronically, SRBP trafficking is already activated. So because you have a lysosome, you have a cholesterol defect in these cells, and then the cells are driving SRBP trafficking to make more cholesterols. And in that scenario, then you can treat the cells with a cyclodextrin to extract cholesterol out of the lysosome to rescue the problem, and then we can cause a reduction of SRBP trafficking. So if you look at these Western blot, where in MPC wild type versus MPC knockout cells, the MPC knockout cell treated with cyclodextrin that rescues the SRBP defect. So you have in the knockout cell, you see more SRBP cleavage, and then you see more phosphorylation of STING, phosphorylation TK1. You see a strong correlation of these activation. Another way we show this is you can take the wild type cell and treat with a variety of different compounds. In this particular case, we use treponemal that will inhibit cholesterol synthesis, then activate SRBP trafficking. When we activate SRBP trafficking, we're also activating STING trafficking, the STING activation. So we think this tether trafficking is really the priming stage of this was activating the STING signal. And some of this work we're working with Joyce Repa to characterize this. And in terms of the lysosome binding or lysosome mediated degradation or the adapter function in PC1, we also did a number of different interaction assays. And one of my favorite is this cell-free based assays. So in this particular assay, we are isolating. So this is really the goal is to show that the STING vesicle is recruited to the lysosome through MPC STING interaction. So we thought we should mimic a STING vesicle and then mimic the lysosome. So we isolated the ER macrosomes from the from a green STING GFP labeled ER microsome and put that on the beads. And then we isolate the red lysosome. So we incubate the green ER vesicles with the red lysosome and see if they will interact with each other. And we're only seeing this recruitment when we use the NPC lysosome. We're not seeing this when we use the LAMP1 lysosome or some other lysosome adapter. So we think that really this is a direct interaction that mediates this contact. And in the physiological cells, this will cause STING degradation. In terms of the boosting of the signaling, then this is a typical biochemical kinetics that we do in the lab to see the STING signaling kinetics. So we can take either wild type NPC1 or NPC1 knockout cells, trigger the STING pathway with the agonist, and then look at the kinetics of either phospho-STING, TPK1, IRFs. So you will see is in the NPC deficient cell, we're seeing a much more long-lasting or more enhanced signaling of the STING pathway. In terms of the protein degradation, this is the total STING protein. When you trigger the agonist, you actually see progressive protein degradation of the wild-type STING, and that's slowed down when you knock on the NPC1. And that also demonstrates this is evolving STING degradation. So the real killer experiment we wanted to do is to see this in the mouse, to see how much STING is really driving the neural pathology in the mouse. So NPC mouse model has been well characterized. These mouse develop cerebellum inflammation, then Purkinje neuronal death, and eventually leading to a motor and coordination defect. So when we compare NPC wild type and knockout mouse, we definitely see reduced body weight. And when you have a STING double knockout mouse, that rescue the body weight substantially. And this is the look at cerebellar ataxia score. This is we do 4 different motor coordination defect and have a composite score that tells you how much defect these mouses are. The NPC knockout mouse has a really severe motor coordination defect with a high score. But the NPC STING double knockout mouse has rescued this quite a bit as well. If you isolate the cerebellum and look at the interferon gene signature, the double knockout mouse also rescued the gene signature quite substantially. And then we also did this just a look at the cerebellum directly. So this is a mouse cerebellum staining with calbidin for the Purkinje neurons, CD68 for actively microglia, and then DAPI for just the nucleus. So in a healthy Purkinje, in a healthy cerebellum, you see these healthy Purkinje cells are mostly red, and you see very little green staining, so there's no microgliosis or microglia infiltration. But in NPC knockout mouse, you lose majority of your Purkinje neurons, and then you have a lot of this activated microglia cells that are in the cerebellum. In the double knockout mouse, we see a substantial recovery of the Purkinje neurons, and then much less of the microglia activation. So we think really deleting STING will see we think STING is really driving a substantial part of the NPC neural inflammation and then neural pathology. We also cross this to IRF and CGAS just to see how much of this is dependent on upstream molecules of CGAS or downstream molecule IRF3. So this similar to what we're seeing in cells, that this is really a CGAS independent process. When you have this is a look at the cerebellar ataxia score, the essential motor coordination defect. When you have a deleted downstream transcription factor IR3, we see a similar rescue as a STING double knockout. And when you delete CGAS, we're really not seeing any changes to NPC disease. If you look at these cerebellum immunohistochemistry, they look the same. The IRF double knockout will rescue some of the Purkinje neurons, but the CGAS knockout does not. So we think really this is a CGAS independent event in terms of the STING activation and STING-mediated pathology. Another thing we think is where is STING expressed in the brain. So STING has been predominantly studied in myeloid cells, in the spleen, in these immune organs. It has never been really looked at in the brain. So we did a antibody staining of STING protein, and then made sure that the antibody is specific compared to STING deficient tissues and then the isotype controls. So what we found is remarkable. These things are almost, this is the STING co-staining with calbedin. This is the Purkinje neuron marker. We're seeing STING predominantly, almost exclusively expressing the Purkinje neurons. And when you do a co-staining with GFAP, this is the astrocyte. We're now seeing almost any expression of STING in myeloid cells. You know, one of the assumption in the field is that the STINGs driving neural inflammation from the glia cells, but we're not seeing STING protein expression in these cells. We think STING prop is likely driving something in the neuronal cells directly. So, you know, giving the model again, we think from this proteomic screen, we actually found the lysosome adapter for STING degradation. And when you don't have this, the MPC1 is this adapter. When you don't have MPC1, we're seeing the SRBP SCAP-mediated tether trafficking to prime the STING signaling through trafficking. We're also seeing a boost of STING signaling because you lose the lysosome adapter, then the STING vesicle accumulate, and then both the prime and boost, we have genetic evidence to show that both prime and boost are important for leading to the eventual neural inflammation and then a neural pathology in the MPC model. And for the MPC aficionados, there's MPC1 and MPC2. So what's interesting is both MPC1 and MPC2 deficiency causes the identical biochemical defect. So it was unclear for a while that why the MPC1 disease in the mouse are so much more severe than MPC2, and MPC1 mutant in the human patients are much more prevalent than MPC2. So about 95% of Niemann pigs are MPC1, only 5% MPC2. So we think that because MPC1 is the lysosome adapter for STING, that MPC2 is the luminal protein. We think MPC2 only gives you the prime, and you still have MPC1 protein, so you're not getting a boost. So that might be the reason why MPC2 is much more healthier than MPC1. So then, you know, I hope this is sort of my summary slide to give you an expanded view of STING. We think that most of the people, you know, unless you're as STING savvy as we are, or as obsessed with STING biology as we are, this is probably what you'll see STING from the majority of the literature. Is STINGs mostly, you know, people think most STINGs active by either DNA or cGAM, and then DNA is going through cGAS or producing this dinucleotide that binds to STING ligand binding pocket. Once STING gets activated, you're predominantly getting type of interferon that will lead to antiviral response, autoimmune disease, DC T cell priming, anti-tumor response. And these are all true, but we think this is only, you know, one side of the STING story. We think there's many other incidences where STING can be activated in the cGAS-cGAM independent fashion. Mostly, we think this has to do with membrane trafficking. We still don't quite understand, you know, how these different membrane trafficking works. But at least in the cases of SAB function mutant or the SRBP tether trafficking, and in MPC deficient cases, we think STING can be activated through trafficking means. And also on the other side, once STING gets activated, you're not only getting interferon, I think you're also getting a number of interferon independent activities. And this list keeps growing in recent years. So there's CellDAT, and James Chin's group have shown there's autophagy, their antiviral response, the tumor invasion. Although we haven't directly shown this, but I have a hunch that these neuron pathologies is probably also interferon independent. So the implication of this is severalfold. One is, I think what's most important is now we know this other side of the STING story. We think this helps with targeted STING therapies, especially with SAVI patients. The SAVI patient, the type 1 interferon therapy for SAVI patients are not very successful. We think that probably has to do with that type 1 interferon is not a predominant driven disease for these STING gain of function patients. So targeting STING itself or some of the downstream effector pathways may be more useful. The MPC patient still will wait and see either interferon or non-interferon pathway are more effective at targeting. but I think STING inhibitor will definitely be a good therapeutic approach. So another implication is STING agonists. So now the major pharmaceutical companies are all developing STING agonists, try to trigger this pathway. What they're aiming for is for a tap interferon response. I think this tap interferon independent response are these consequences that it's also important to consider when you activate the STING pathway, either in a tumor microenvironment or systemically. So another thing is this traffic-based STING activators. We think, as we're seeing more and more examples of this membrane trafficking-based STING activation, this could become a therapeutic design. We could think about different ways that we can trigger this membrane trafficking-based activation as a different way to act this. One of the reasons being that one of the limitations of the ligand-mediated activation is there's a common variance in the ligand binding pocket of the STING gene that's causing some of the challenges for the STING agonist. So I think the trafficking-mediated activation will be a good solution for that. And then, this reminds you that the interferon is really a more recent evolutionary event for STING biology. The majority of the STING proteins are actually evolved from millions of years ago. So, we think this interferon-independent activity, the list of this activity could grow as we investigate this more. So with that, this is people in the lab. The JJ is the one that did the interferon-independent 365 mouse. He now has his own lab in Cleveland Clinic. And Ting Ting and Xintao are the two people in the lab that did the proteomics screen I talked about. The Ting Ting have been most working on the NPC story that's recently accepted in Nature. And then Xintao has worked on other haste of the trafficking that we think is also very interesting in terms of regulating STING activities. Jane Leo helped with some of the early T cell studies, and Joyce Ripa has been very, has helped us quite a bit with the NPC mouse models, therapeutic models, understanding the NPC biology, SRPP biology. So I really thank both of them for their help. And also, Yang-Xin and Alex, my go-to tumor cancer experts for tumor studies. And other, also thank my funding sources. I'll thank you for your attention.
Thank you, Nan, for your wonderful talk. So I'm sure that will also trigger a lot of questions. So Nan, yeah, okay, let me let DJ to ask the first question.
You first. Can you hear me?
Yes.
Yeah. Okay. So now that was a beautiful talk. I have a question about these interferon-independent activity of STING. So what do you know about those? I mean, what are the downstream mediator? I assume it's not IRF3, right? It would perhaps be independent IRF3 because IRF3 is not conserved in those other species, right? You mentioned. So what do we know about?
We don't know much. And the one we know the most, apart from the studying the T cells. So actually I have a line drawing here. So one of the domain we mapped is what we call the UPR and trafficking motif. So if you look at the three-dimensional structure, so there's a helix on the side of the STING structure that's floating off the structure. And then that's the domain I think we mapped to for at least mediating a T cell death. We think that is at least on the STING side, that's regulating interferon-independent activities. Now, how do STING activists, what's the cascade, and that we don't know yet. And this could be different in different cell types. We think in the T cells, it could be leads to T cell death. In other cell types, it could be driving other different effector functions.
Okay, second question by Peter Lee.
Hi, Jan. That was a fantastic talk. I have a question regarding the tumor cells in the microenvironment that can potentially activate different immune cells. So is there evidence that the molecule that's doing the activation or transfer is actually CGAMP, or could it also be double-stranded DNA? Do you know anything about that?
Yeah, I think there are evidence for both. There are definitely evidence for CGAMP. There people have done work where they take either CGAS positive, CGAS negative tumor, then take CGAS positive or CGAS negative immune cells, and to show that the CGM is produced in the tumor and received by STING in the neighboring immune cells. So that has been genetically shown. There are different ways this can be passed on. There are cell-cell gap junctions this can pass on. There are, there are secretions can pass on. So CGM, CGM, even people promoting this as an immunotransmitter, and just involving short-range transmission from tumor, a short-range communication between tumor and immune cells. Now, the predominant literatures on, DCs on NK cells that give you anti-tumor response. So we think this also happens with T cells, which will end up to be a pro-tumor effect.
So Nan, yeah, let me ask one question. So, so you nicely showed that MPC1 served as a degradation receptor of STING in the lysosome. So is that cell type dependent? Is this does this happen in T cells, for example?
We I don't know if it's cell type dependent. The degradation experiments are mostly done in fibroblasts. We think at least, in vitro, we've been looking, it happens in fibroblasts. In vivo, we don't know what cell type it happens in. In vivo,
but you kind of implied in vivo it may happen in Purkinje neurons, right?
Yeah, we think, one of the challenges with study Purkinje neurons, we can't really pull these cells out and then look at them. So, we don't really know if this is happening in Purkinje neurons. I think the evidence has been that Purkinje neurons being the driven causes for disease, NPC disease, and STING is exclusively expressed in Purkinje neurons. But we need a way to culture Purkinje neurons in the tissue culture to be able to do all the biochemistry, do all the molecular analysis. Yeah.
Okay. So DJ, do you have another question? Okay. Okay. Let me see. Next one. Oh Yang, please.
Hi, can you hear me?
Yeah.
Hi, Nan, beautiful talk. Thank you. I have a question. In one of your slides, you mentioned that the STING mutant, N153S mutant, you believe that it caused ER stress, and this ER stress mediates its pathological function. So you showed BIP and CHOP, which is beautiful Western, very convincing. I wonder whether you have looked at IRE1 XBP1 pathway. And what did you find, if so?
I remember we might have. I think we spent quite a bit of time looking at what is causing the ER stress. I think the conclusion was, I think we had looked at the different, the IRE1 pathway, PERK, and different ER stress drivers. I don't know what the conclusion is. It's probably not, each one of these drivers specifically. I think the molecular defect we think start from calcium release. One of the hypothesis we had is because STING translocates from ER to Golgi, and then when STING leaves ER, it's actually a fairly massive event. There's a large amount of STING protein all leaves ER in an instant. We think that causes ER calcium leakage. And then Jinliu has helped us to measure that. So there's some calcium problem with STING trafficking. We think that may be the initial trigger for this ER stress. I don't remember a specific requirement for either IRE1 or PERK or each one of these pathways.
All right, thank you.
Sure.
Okay, so because of the time, let us ask Jin Jiang, ask the last question. Jin, are you there? Yeah, Jin just dropped off. Okay, let me ask one more question. So, so you showed multiple types of autoimmune disease, maybe triggered by the overactivation of the STING pathway. However, you only showed in one disease that you observe the T cell cytopenia, but not in other type of autoimmune disease. So you mean in the other type of autoimmune disease, the T cell are not kind of be triggered for over-apoptosis?
Yeah, that's the thing, we don't quite understand. And then in the TRAX1, for example, so TRAX1 deficiency causes chronic activation of C-gaspine, right? And then in that mouse, then in those humans, and then T cells not a major problem. So we don't know why. I think, one of my personal favorite hypothesis is maybe when you have a ligand-mediated activation, you're predominantly getting interferon and some interferon-independent activities. But when you have a ligand-independent activity, this memory-mediated activation, maybe you're predominantly getting interferon-independent activities and then less interferon-dependent. Maybe depending on how you activate STING, then you sort of gear towards either interferon or non-interferon outcomes. We don't know how the protein decides that, in what scenarios. So that's definitely something worth looking into. But that's definitely true. And a lot of the sort of canonical DNA-mediated autoimmune disease, that T cells not the T cell doesn't have a major problem, and they don't develop lung disease either. Versus in the membrane trigger STING activation, you get, I think not just SAVI, there's a more recent one, COPA disease, also chronic active STING, independent C-gas, that also causes the lung disease and then a T cell problem. So there maybe just depends on how the protein gets triggered, then it will give you different effect.
Thank you, Nan, so much. So yeah, because of the time, it's already 3:03. So yeah, we'll stop here. So if you guys have any additional questions, you're welcome to stay to ask more. So yeah, thank you all.
Okay, thank you.